Share your screen, and we should be live on air right now. So we are a minute early, but we can start a longer introduction. To thank Professor Jetten for doing the micro seminar today. So Mike Jetten is a world-renowned professor for anaerobic microbiology, and we're very excited for him to share his discovery of impossible microbes. And so recent publications and recent discoveries have pushed the field, I think, even further in terms of what we can discover, and we will hear updates from Mike today. And so, Mike, if you'd like to take it away and share your screen, and I'll remind everyone that we will ask questions through Twitter using the hashtag #USeminar, and if you're inside the Hangout afterwards, we can ask questions, and mute yourself during the talk, and then we'll unmute you at the end of the seminar. All right, so Mike, if you want to take it away.
Thank you very much, Jenny, for this nice introduction and being part of micro seminars.
Yeah, looks good.
Looks good? Okay. So, ladies and gentlemen, it's a great pleasure to share with you the discovery of impossible anaerobic microbes in today's micro seminar. I'll start off with a brief introduction into anaerobic microbiology and then share with you several of our recent examples. Anaerobic oxidation of ammonium. We'll cheat a little bit on the oxygen for complete ammonium oxidation by ANAMMOX. I'll share with you some ammonium oxidizing symbionts, and then as the last example, we'll move on to anaerobic oxidation of methane. Well, I'm probably preaching for our own parish, but the Earth is a microbial planet. There are many, many very useful anaerobic microbes. They treat our water, they make our water clean. They produce the oxygen we need to breathe. They fix nitrogen for our crops. Very important, they make delicious fermentation products, and they help us digest food, produce essential vitamins, and are important for making new medicines and discovering new antibiotics, and they help us clean up our xenobiotic spills. In contrast to common belief, there are only very few pathogens. A recent very nice paper by Jos Neufeld and Jack Gilbert showed that life without bacteria is possible, although probably very miserable. But if we ask the question, how much do we actually know about the microbes on our planet? If we look at culture collections, we are able to cultivate about 40,000 different stains in the laboratory. If we look at the latest release of the ribosomal database project, we see that we know the identity of more than 3 million 16S RNA ribosomal genes. But if you look on Earth, there are many, many more microorganisms out there. And that's good for us as microbiologists. There is a lot to discover. If you look at this pie chart, there is a lot of terra incognita that we can discover and explore. And within the Center of Anaerobic Microbiology, we try to find new anaerobes that would be good for our health, would be even better for our environment, and help us make a sustainable bio-based economy. Well, you cannot study all microorganisms. So many of you might study organisms harvest the energy from the sun, phototrophs. I'm certain that very, very many microbiologists study chemoorganotrophs, so heterotrophs that convert organic molecules. And during my career, I have specialized in microorganisms that used small inorganic molecules like hydrogen, hydrogen sulfide, ammonium, and use that as an energy source. These are called chemolithotrophs. Most of them are also autotrophs, so chemolithoautotrophs. When we moved from Delft to Nijmegen some 15 years ago, we made this, let's say, periodic table of impossible anaerobic microbes. If you look at the upper row of this periodic scheme, then you see the energy-rich compounds, the electron donors, hydrogen, methane, hydrogen sulfide, ammonium, and on the first column you would see a redox tower as microbes could encounter them in a sediment. So the top layer of the sediment, oxygen, would be available, but would be rapidly consumed in the first few millimeters, and then the organisms are left under anoxic conditions with alternate electron acceptors. They would encounter, for example, nitrate, iron, in marine systems sulfate, and if nothing else is left, they have CO2 as the ultimate electron acceptor, either producing methane or acetate. You can see that by each of these reaction combinations, there's in principle enough free energy that microorganisms could live and thrive on it. And with the green boxes, we more or less know organisms that can do the job. But there were at least two very conspicuous boxes. That is the box of organisms converting methane at the expense of nitrate, or the box of organisms that could use nitrate to oxidize ammonium. Many of the colleagues have been looking for these microorganisms, trying to enrich and isolate for them, and all these attempts were in vain. So slowly in the literature they got the name impossible microbes. Well, these microbes have probably been around for 3.5 billion years, so the impossible is maybe more our capability to find and grow them than on the organisms themselves. Nothing is impossible, especially if a wise and unfortunately lately deceased person, Nelson Mandela, said it always seems impossible until it's done. And so we took up the challenge to find these impossible microbes, not by doing the same things as had been done before, but by designing another and new strategy. Now, for those faculty members who have to teach, like Jenny, or are otherwise occupied, there is a 2-minute summary on YouTube, but for the rest, please bear with me for the next 40 minutes and I'll explain the story in more detail. So what is our strategy? What is our vision to find these impossible microbes? First of all, it's good to have biogeochemical colleagues or aquatic ecologists as colleagues who can, with whom we can discuss interesting ecosystems. Ecosystems where there is a limited amount of oxygen and where there is plenty of ammonium, methane, and nitrate. What we then do is try to bring the best samples to the laboratory. With our engineering background, we try to design and operate bioreactors under optimal conditions and set up enrichment cultures under the best conditions, which mostly means starvation and substrate limitation. With some patience, you can grow enough cells, and once you have enough cells, nowadays you can unleash the molecular toolbox to unravel the secrets of these anaerobic microorganisms. With the knowledge we have gained, we can go back to the ecosystem to see if we indeed enriched for the important organisms. And as microbiologists, we keep a keen eye on the applications of these microbes in anaerobic waste water treatment or in anaerobic gas treatment. You need a few things to employ this strategy. First of all, I'm very proud and privileged to work with a fantastic team of excellently educated and enthusiastic team members. And if I may do a little ad, there are still some vacancies, and we have a very nice master program in microbiology in which you could join us. Further, the questions we want to answer are so complex that you never can do that that you not can do it on your own, and it's very nice and rewarding to collaborate. Both nationally and international collaboration is necessary to tackle these questions. And we are very happy to have the Jetten Institute to stimulate this collaboration in anaerobic microbiology. It's not only for the people who participate, but also for the audience. Please be aware that we have SIAM talent grants. Actually, one of the students from Waterloo University is joining us at the moment with such a talent grant. And also for the senior staff, we have visiting scientist programs so that we can host you up to 3 to 6 months. In addition to collaboration and staff, you need, of course, state-of-the-art method. And in our case, that is bioreactors, and more bioreactors. To enrich the impossible anaerobic microbes, then, of course, nowadays we apply metagenomics to the systems to unravel their blueprints. And once you have the blueprints, you can design much more dedicated experiments to unravel their secrets. All right, after this brief introduction, we'll move on to the examples, starting with anaerobic oxidation of ammonia. For a very long time, this was how microbiologists viewed the nitrogen cycle. In 1899, Martinus Beijerinck, one of the first professors of microbiology in the Netherlands, discovered nitrogen fixation, in which dinitrogen gas is fixed into ammonium. Then by the works of Sergei Vinogredsky, we knew that ammonium can be oxidized to nitrite by ammonium oxidizing bacteria. Nitrite can be oxidized to nitrate by nitrite oxidizing bacteria. And then nitrate can be reduced all the way back via nitrite, nitric oxide, nitrous oxide to dinitrogen gas. In the 70s, there was a nice paper by the theoretical biologist Engelbert Broda. He wrote a paper on the two lithotrophs missing in nature. And as you can see from the calculations, one ammonium converted together with one nitrite yields dinitrogen gas, and there is sufficient free energy in this reaction that organisms should be able to thrive. As a small anecdote, apparently as a theoretical biologist, you have some time on your hands, and Engelbert Broda was moonlighting, as this very reliable Daily Telegraph mentions in its report. Some 20 years later, Arnold Mulder, the chief wastewater engineer at Gest Brocadis, a yeast and penicillin producing factory in Delft, the Netherlands, he was treating the sulfate-rich wastewater of that plant in an anaerobic system. And if you have sulfate-rich wastewater in an anaerobic plant, you eventually will end up with hydrogen sulfide. Of course, the citizens of Delft were not very pleased with the smell of this sulfidic hydrogen sulfide, and they complained. Arnold being a very pragmatic engineer, he bought a lot of calcium nitrate. He added this calcium nitrate to his anaerobic wastewater treatment plant, and inadvertently he had created the ideal conditions for ANAMMOX to thrive. Luckily, he was very careful in measuring the various compounds in the wastewater, so he measured the ammonium in the influent. He also measured the ammonium in the effluent, and within half a year after the additions of calcium nitrate, he saw that ammonium was disappearing from an anaerobic wastewater treatment system. Then Arnold came to us at the TU Delft at that time and asked us to investigate the biology and microbiology of the system. And that was when the first PhD student on ANAMMOX, Astrid van der Graaf, could show that this was really a microbial process. To make a very long story short, the first thing we tried to do in the research was to get rid of the pilot plant. It was a very pain in the neck to operate, and Mark Strauss and Seth Heinen designed a sequencing batch culture fully dedicated to the growth of ANAMMOX bacteria. We still use these systems in the laboratory to grow enough biomass of the ANAMMOX bacteria, and I have 3 short movie clips to show you how ANAMMOX reactors look in real life. First, the reactor lab with several of these ANAMMOX systems in operation. Then you see the beautiful red ANAMMOX biomass. ANAMMOX reactors run for a long time, but they cannot do without maintenance, as you will see in the next movie. And although ANAMMOX biomass grows very slowly, you periodically have to remove biomass from the reactors to keep the aggregates in the reactor alive and running. So if you see a bleed bottle in the next movie clip, that is to remove the biomass once a week from the reactor system. So with these dedicated systems to grow enough ANAMMOX biomass, we were able to identify the ANAMMOX bacteria as planktomycetes. And the first ANAMMOX bacteria were called Brocadia, after Hist brocades and their beautiful red color. We have the Cunenia ANAMMOX bacteria in Germany and Switzerland, thriving there in wastewater treatment systems. We have the marine ANAMMOX bacteria called Scalindua. And some 8 years ago, I was very much honored by a group of Chinese and Korean scientists who named one of the ANAMMOX bacteria they discovered in Asia after me. Together with Jaap Zinnige-Damstee at the Royal NIOS, we were able to identify that ANAMMOX bacteria have very unique laddering lipids with a kind of staircase structure. These laddering lipids are unique biomarkers for ANAMMOX. If you think about ANAMMOX bacteria and ecosystems where they could thrive, then the oxygen minimum zones in the ocean are one of the systems that come to mind. In these systems, there is ammonium from biomass degradation. A lot of the oxygen has been consumed by decaying biomass. And together with Marcel Kuipers from the Max Planck Institute in Bremen and Jaap Zinnige-Damstee from the NIOS, we sailed the 7 seas and investigated marine ANAMMOX bacteria. In the next map, you can see the many different places that we have visited, starting in the Black Sea, later on in the Namibia upwelling system here on the coast of Southwest Africa, Peru and Chile oxygen minimum zones, and recently the Arabian Sea. On all the stations that we have visited, we could show the activity of ANAMMOX bacteria with stable isotopes. We could see the presence of their genetic material in the form of 16S RNA molecules. We could do fish microscopy, and we could use the diagnostic laddering lipids to show that they are really present in these systems. If we do the gross calculations, it might even be that the ANAMMOX bacteria are responsible for 50% of the nitrogen loss in these ecosystems, and thereby significantly contributing to the loss of nitrogen from marine ecosystems. The list continues with different ANAMMOX research that we have done in the past years. Of course, the elucidation of the first blueprint of Cunenia stuttgartensis was a great breakthrough for us. Now we could really see what the genetic makeup of the ANAMMOX bacteria was. Lara van Niftig has been doing some great cell biology on the ANAMMOX organism, not in the least elucidating that they have an internal organelle, the anamoxosome, where all the cytochrome C proteins are located that are involved in the ANAMMOX metabolism. We could show that the rocket fuel hydrazine and nitric oxide are important intermediates in this process. And in addition to marine ecosystems, together with Gui Bin-Su of the Chinese Academy, we could also show that in at least 80% of the Chinese wetlands that we investigated, ANAMMOX plays a significant role. And recently, together with Thomas Beirens in Heidelberg, we could elucidate several of the protein structures, and Dan Speth was recently able to elucidate the full metagenome of an ANAMMOX wastewater treatment system. To make a long story short, here we have the ANAMMOX pathway as we think it operates today. If we start with nitrite, nitrite has to be reduced by a nitrite reductase to the level of nitric oxide. And here, similar to denitrification, there is some diversity in the systems that ANAMMOX bacteria use to reduce the nitrite. So for an example, the marine ANAMMOX bacteria, they use a iron-dependent CD1 nitrite reductase, NIRS. In Jettenia, for example, there is a copper nitrite reductase, NIRK. And in Brocadia, there was a recent paper from the Satoshi Akabis group that it is yet unknown which systems Brocadia ANAMMOX bacteria use to convert nitrite. Then nitrite is combined with ammonium by an enzyme system that is called hydrazine synthase. The hydrazine produced is subsequently oxidized by hydrazine dehydrogenase, yielding the 4 electrons needed to reduce nitric oxide and NO. Over the recent ANAMMOX findings have been highlighted in the on internet and in the press with very interesting Titles: Caturine, turn power to space travel, P-power, nitrogen cycle to the moon, rocket fuel, saving the world, saving Mars. Very interesting stories about how ANAMMOX and hydrazine could be used. So for us, it was very intriguing to find out how indeed ANAMMOX bacteria can make the rocket fuel hydrazine. And for that, we took the most pragmatic and simple approach. If hydrazine is an important intermediate, then probably there is an important gene and gene product necessary to make hydrazine. And for once, it worked out. Indeed, when we looked at the transcriptome of ANAMMOX bacteria, we see a 3-gene cluster that is very highly transcribed. And we could also purify the gene product as the hydrazine synthase with the 3 subunits, the big subunit corresponding to the large gene and the 2 other subunits. About 20% of the ANAMMOX protein consists of this hydrazine synthase. Together with Thomas Weyrenscoop in Heidelberg, Andreas Dieter was able to crystallize the hydrazine synthase, and then Wouter Maalke had the dubious task of getting these protein preparations to work. With a lot of trial and error, we eventually succeeded in using a combined assay, in using both the hydrazine synthase to produce the hydrazine, and then in combination with the hydrazine dehydrogenase to convert the produced hydrazine into labeled dinitrogen gas. And although the activity is not as high as we wish it to be, we can clearly see that ammonium and nitric oxide are ultimately converted to dinitrogen gas via the intermediate hydrazine. We were also able to make antibodies against hydrazine synthase large subunit, and when we use these hydrazine synthase A antibodies, we could show that the hydrazine synthase enzyme complex is located in the anamoxosome of the ANAMMOX bacteria. Of course, we have still a keen interest in applying the ANAMMOX process for wastewater treatment system, and in this slide, a cartoon how that road to full-scale application came about. So after the discovery of ANAMMOX bacteria in oceans and wastewater treatment, we took out a patent application together with the Foundation for Applied Sciences. But a patent as such is not worth very much if you don't have an industry or a customer that wants to work with it. So we have been very fortunate to work with a company, PAC, who took a license on this patent and invested enough time and money to bring it from a license agreement in 4 years to a full-scale ANAMMOX wastewater treatment plant in the city of Rotterdam. It took quite a while by trial and error to figure out how an ANAMMOX full-scale operation works, but since 2006, ANAMMOX is being exported to the Far East to treat the wastewaters there, and also now recently both the USA and Brazil have opened their markets to wastewater treatment markets to be treated with ANAMMOX systems. All right, by identifying the ANAMMOX bacteria, we have been able to shortcut the nitrogen cycle. So from ammonium, a direct combination with nitrite, ANAMMOX bacteria shortcut this into dinitrogen gas, and hydrazine synthase, hydrazine dehydrogenase, and nitrite-nitrate oxidoreductase are good enzymatic biomarkers for ANAMMOX in this nitrogen cycle. Then the question, of course, remains, how is ANAMMOX supplied with nitrite? And it does this in cooperation or competition with other nitrogen cycle microbes. ANAMMOX bacteria can operate with aerobic ammonium oxidizing bacteria, with aerobic ammonium oxidizing archaea. We think they are in competition with nitrite oxidizing bacteria under oxygen limitation and nitrite limitation. When there is sufficient nitrate and electron donor, denitrifiers could make nitrite for ANAMMOX bacteria. Also, organisms that are able to do decimatory nitrate reduction to ammonia could supply ANAMMOX with nitrate and ammonia. And as we will later on see in the micro seminar, anaerobic oxidizing methane archaea could convert nitrate with methane into nitrite for ANAMMOX bacteria. In the next slide, I've summarized the different studies we have done to show that indeed ANAMMOX bacteria can cooperate with others to convert ammonium with nitrite into dinitrogen gas. The earlier studies of Olaf Slickers on the high ammonium concentration and low oxygen concentrations give very nice consortia of aerobic ammonium oxidizing bacteria together with ANAMMOX. Jetten has done some elegant studies on the marine conditions where we see that low ammonium concentrations together with low oxygen give consortia of archaeal ammonium oxidizers together with ANAMMOX. Lena Ross has done some nice studies where she could show that under sulfidic conditions, DNRA bacteria provide nitrite to ANAMMOX bacteria, or under low oxygen, low nitrite conditions, you establish consortia of denitrifiers and ANAMMOX. And early on, we and others have shown that under high methane and no ammonium, no oxygen concentrations, ammonium oxidizing archaea or bacteria can cooperate with ANAMMOX. I would like to draw your attention to the most recent investigation that we have done, and that was when we stumbled upon a collaboration between Nitrospira and ANAMMOX bacteria on the low nitrite and low oxygen concentration. Sometimes you find things that you were not really looking for. Also here, it started with a theoretical paper by Costa Perez and Kraft, who wrote a very interesting article on the labor division in nitrification. As I said already, for some 100 years there was a dogma that nitrification was split between two different groups: the aerobic ammonium oxidizers that convert ammonium into nitrite, and the aerobic nitrite oxidizers that convert nitrite into nitrate. And then, of course, the question was, why is there no organism that can convert ammonium directly into nitrate? The story started a little bit more different. Maartje van Kessel, one of the PhD students in our ERC ANAMMOX project, was given the task to find new ANAMMOX bacteria that we had seen in a aquaculture biofilter. So she set up a bioreactor dedicated to find new ANAMMOX bacteria that she fed with water from a aquaculture system and gave very low concentrations of ammonium, nitrite, and nitrate. She made sure there was no oxygen and no additional carbon source other than the carbon that was in the fish water. So after a year, she saw very good consumption of ammonium and nitrite in the system. And when she did fluorescent in situ hybridization with a probe in pink for ANAMMOX, she saw, of course, that 40% of the biomass were ANAMMOX bacteria. And if she used a probe specific for Nitrospira bacteria, she saw that there were very nice aggregates where Nitrospira and ANAMMOX were together. But it was very intriguing then to find out what these bacteria were actually doing together. Were they cross-feeding? Were they giving each other intermediates? And what was Nitrospira doing? Was Nitrospira really oxidizing nitrate? Or was Nitrospira more versatile? Was it maybe converting nitrate into dinitrogen gas? So to find this out, we took a metagenomics approach. We extracted the DNA from this culture system. The DNA was sequenced by ion torrent technology and Illumina sequencing. The contigs were assembled and the genomes were binned out. And of course, we analyzed then the genomes, and on the basis of genomes, we could design new physiological experiments. So Mats Albertsen of Aalborg University was able to do a very nice binning and get two very high quality draft genomes of Nitrospira out of here. Here you see the bins in this picture. And when Sebastian Lücker then analyzed these nearly complete genomes, we were very surprised to see that on the contig, which had the diagnostic NXRA and NXRB genes, so the nitrite oxidoreductase, we could also find the AMOC A B operon, encoding for the particulate ammonium monooxygenase, and also found the genes for hydroxylamine oxidoreductase. Of course, when we saw these genes, we could design new experiments. We could indeed show that when you do the ammonium oxidation experiments in the presence of allyl-thiourea, that no ammonium was ever converted, while nitrite oxidation continued. Also, by doing a combination of micro-autoradiography and fluorescent in situ hybridization, we could show that the Nitrospira cells were taking up radioactive bicarbonate, and by using very specific fluorescent components that are suicide substrates for ammonium monooxygenase, we could very specifically label the Nitrospira cells. In addition to being very exciting research, this is also an excellent example of synergy and collaboration. While we were working at the process of complete ammonium oxidation, we became aware that our colleagues in Vienna were also working on this process, and we synchronized our studies and could show in independent research that indeed in both systems Nitrospira were responsible for complete ammonium oxidation, and that both systems have very unusual and novel AMO-A genes that are not captured by the genes probes available at the moment. Our future research is directed at isolating the common MOX bacteria and making very defined co-cultures to see what else in the nitrogen cycle they could do. And also we have several studies dedicated to the environmental relevance and detection of these organisms. This aquaculture system gave also rise to a second surprise. While we were investigating the aquaculture, we also got interested in the interaction of the carp in this aquaculture system with their microbiomes. And if you think of fish, they mostly excrete the nitrogenous waste as ammonium. So if you think that the gills of these carps are the place where they excrete the ammonium, this could also be a very good place to look at the microbes which would be living in and on the gills of these fish. So also, to make a long story short, what we could show is that in the gills of these carp there are nitrosomonas bacteria residing that convert the ammonium with oxygen to nitrite, and that also in these gills there are aerobic denitrifiers, beta proteobacteria, probably pseudomonas, that under aerobic conditions can convert the nitrite into dinitrogen gas. Marge was able to prepare the gills and put them into small incubations, and we can see that both fish and prepared gills, if you incubate them with nitrite but no oxygen, you hardly see any labeled dinitrogen gas. When you incubate them with labeled ammonium but no oxygen, there is hardly any labeled dinitrogen gas. But when you incubate fish, or when you incubate prepared gills with 15 ammonium and oxygen, there is a sizable production of 29 30 dinitrogen gas, and also the aerobic denitrifiers can convert the nitrite into dinitrogen gas. And then Rob Messman and Marge together did very nice, sophisticated fluorescent in situ hybridizations and electron microscopy to indeed show that the nitrosomonas and pseudomonas cells are present in the gill of these carp, and also that there are actually endosymbionts of the fish that are responsible for up to 30% of the ammonium conversion excreted by these fish. From the nitrogen cycle, we now move on to the anaerobic oxidation of methane. Methane as a compound was discovered already some 300 years ago by Alexandro Volta, and the importance of methane in the environment and for our households was also recognized early on. Here you see a picture of John Dalton collecting marsh fire gas as an important greenhouse gas in these systems. And here you see the importance of methane for the Netherlands. A new statue recently unveiled by our Queen, celebrating 50 years of gas extraction in the north of the Netherlands. If you want to see the importance of methane, of course, we know that wetlands are very important methane sources, but luckily we have the aerobic and anaerobic methane oxidizing microbes that take away much of the methane before it escapes into the atmosphere. As a movie interlude, I have here a nice YouTube clip for you. So you can see that in this Minnesota lake in winter, the methane is trapped under the ice. When you make a small hole in the ice, the methane can escape, and when you lighten this methane, you get a very nice flame out of the ice. So as already said, aerobic methane oxidation is already known for 110 years. Also, the name giver of our institute, the Söhngen Institute for Anaerobic Microbial, Nicolaas Söhngen, he did his PhD at Delft University investigating the origin and conversion of hydrogen and methane. And recently we got more interested into the anaerobic part and finding the microorganisms that use nitrate, nitrite, or even iron for the conversion of methane in the absence of oxygen. For that, you have to go to ecosystems where there is an abundant supply of nitrate, either from agricultural runoff or from groundwater, and a high methane production in the sediment. We have sampled several wetland systems in the Netherlands, and also recently we visited rice fields in Italy or the Botnian Sea in the north of Europe. Under the ideal conditions, you'll make a counter gradient profile of nitrate and methane. In this slide, you see a typical, which of course means the best profile we ever made. If you look in the profile, we measured in red the methane in the pore water and in green the nitrate in the pore water. At about 160 centimeters, you see a steadily increase in the methane concentration in the pore water, reaching a maximum at about 120 centimeters, and then you see a steep decline in the methane concentration of the pore water, and actually no methane ever reaches the oxy zone in this ecosystem. And the place where you see the steepest gradient of methane, there is also the steepest gradient of nitrate consumption. And of course, this would be an ideal niche for nitrate-dependent anaerobic oxidation of methane. So we took the sample back to the laboratory, and then of course use different tests. We use activity tests. You can use qPCR if you know which organisms you have to look for. You can use stable isotopes 15N and 13C to investigate the samples. You can try to enrich for the responsible microorganism, do fluorescent in situ hybridization, and also do a metagenome to see what these organisms are up to. Here are most recent activity tests performed by Annika Vaxma in soil slurries. So she made 50 milliliter soil slurries, supplied them with 13C labeled methane, and then when there are no additions, you see no increase in the 13 CO2 percentage in the gas phase of this incubation after 20 days. When you only add labeled methane, you see a very small decrease, indicating that there are still some endogenous electron acceptors in the system. In this case, when you add 1 millimolar nitrite, you see that there is a small increase in the 13CO2 production. But when you add 5 millimolars of nitrate to this soil slurry, then you see there is a significant increase in the 13CO2 concentration, indicated that indeed within 20 days anaerobic oxidation of methane occurs. If we would wait longer, another 20 days or even another 40 days, then also the nitrite-dependent methane oxidation would kick off. And if you know what organisms you would have to look for, then you could do a qPCR or an AOM archaea or bacteria in the system. So Annika also took the paddy field soil to start up an enrichment cultures, giving nitrate as the electron acceptor, and then following, of course, the enrichment with qPCR and nitrite and nitrate measurements. Well, this is not trivial. Although we can see activity of the nitrate-dependent microorganisms within 20 days, it takes a long time before these organisms really kick off and start to grow in the enrichment systems. But with enough patients, you ultimately can see co-cultures of anaerobic oxidation, anaerobic methane oxidizing bacteria and archaea. We have done that a couple of times. And when you use nitrate as an electron acceptor in these systems, then you end up with consortia of archaea and bacteria. And when we first did it in 2006, it was one of the times that we made the 8 o'clock news and had our 30 seconds of fame. The 8 o'clock news in the Netherlands is in Dutch, but at least I'd like to show you how a microbial item on the news in the Netherlands looks like.
Het is een mirakelse bacterie, ontdekt in het Twentekanaal en mogelijk inzetbaar tegen het broeikaseffect. Onderzoekers van de Universiteit van Nijmegen hebben de bacterie gevonden. Die eet methaan, een broeikasgas dat nog schadelijker is dan het algemeen verguisde koolstofdioxide. Methaan is afkomstig uit bestrijdingsmiddelen die in de landbouw worden gebruikt.
Dit zijn de bacteriën. Al een eeuw vermoeden wetenschappers dat ze bestaan. Nog niemand had ze ontdekt. Een onderzoeksteam microbiologen uit Nijmegen heeft ze gevonden. Het heeft een jaar geduurd voordat andere wetenschappers ervan overtuigd waren dat deze nieuw ontdekte bacteriën methaaneters zijn.
By now we know that when we feed these consortia of archaea and bacteria with sufficient nitrite, that the bacteria like the nitrite much better, and that we can further enrich the Methylomirabilis bacteria. And in the middle, I'll show you that they possibly have a NO, nitric oxide dismutase system. When you feed more nitrate to the system, the archaea seem to prefer the nitrate much more than the nitrite, and they have been recently given the name methanopyridines nitroreducens. If we look at the Methylomirabilis microorganisms, they also show up beautifully in electron microscopy pictures with a polygonal shape. As many of the organisms that we study, they are slow growers with doubling times of 2 to 3 weeks. At the moment, Karen Stultjes, within a project of the Foundation of Applied Sciences, is looking at the ecophysiological parameters. How good are these organisms in removing nitrite? How good are they in removing methane? And how much biomass can they make at the expense of methane and nitrite? And in addition to consuming methane, Olivier Razigraf was able to show that these organisms are also autotrophs, that they use the Kelvin cycle to fix the CO2. This also means that if you do environmental incubations with 13 methane, it will not be assimilated by these organisms, and that you have to add 13 labeled CO2 to find them. Together with the French Genome Sequencing Institute Genoscope, we were able to elucidate the blueprint of Methylomirabilis oxyfera, and very much to our surprise, we saw an aerobic pathway for methane oxidizers. So in the genome, we could see the PMO CAB operon encoding for a particulate methane monooxygenase and a methanol dehydrogenase. And we could not find a complete denitrification pathway. Yes, there was a nitrite reductase that could convert nitrite into nitric oxide, but there was no canonical nitric oxide reductase and no canonical nitrous oxide reductase. So on paper, it is then possible to make an hypothesis that these organisms must have a putative nitric oxide dismutase that converts the nitric oxide into dinitrogen gas and oxygen that subsequently can be used in the PMO reaction to convert methane to methanol and ultimately to carbon dioxide. Well, more easily said than done. This, of course, needs experimental validation. And for that, we had to tease Methylomirabilis in several ways. We made use of suicide substrates for the MMO, for inhibitors of the MMO, and of dual-labeled nitrite to find out what these organisms were really doing. So in the setup, the Methylomirabilis cells were fed with dual-labeled 15N18O nitrite. We gave them formate as an electron source that they could push forward the reaction, converting nitrite into labeled nitric oxide, and then the nitric oxide would dismutate into dinitrogen gas and oxygen, and by inhibiting the methane monooxygenase with acetylene and or using propylene as a suicide inhibitor, we hoped that we could get the cells to release some of the oxygen that we could then measure with very sensitive mass spectrometers. After several tests, Katrina Edwig was able to show that indeed when you treat the cells with the propylene and with acetylene, that you inhibit the methane monooxygenase enough so that some nanomoles of oxygen are indeed released in these systems. So much for the Methylomirabilis bacteria. Recently are more focusing on the archaea. Also there we have taken the metagenomics approach, extracting the DNA, constructing draft assemblies, and looking into the physiology and biochemistry of these organisms. So what we can show is that the Methanoperidans archaea are really capable of converting the nitrate into nitrite. So once in a while we see a small accumulation of nitrite in the systems. The Methanoperidans archaea are always enriched together with nitrite scavengers, and the nitrite scavengers can either be the Methylomirabilis bacteria if there is sufficient methane in the system, or when there is sufficient ammonium, they can also be co-enriched with ANAMMOX bacteria. And if the Methanoperidans archaea have to do the job on their own, they will reduce the nitrate all the way to ammonia. This is a recent fluorescent in situ hybridization picture taken by Simon Guerrero, showing that you have indeed a lot of Methanoperidans archaea in the enrichment culture. Arslan Arshad and Cornelia Welton have looked deeper into the metagenome of this AOM archaea. What we can see is that there is a complete reversed methanogenesis pathway present, that there are several respiratory complexes in the encoded in the metagenome of these archaea, that they have a peculiar BC1 complex, and that there are many genes encoding for cytochrome C proteins, and of course that they have the genomic inventory to convert nitrate into nitrite by an archaeal nitrate reductase, and they have the inventory a pentaheme cytochrome C nitrite reductase that can convert the nitrite into ammonium. And for the moment, we are looking for a very skilled master student to become a PhD student on the biochemistry of these archaea. In January, nitrate is a much more abundant electron acceptor in freshwater systems than nitrite, so we expect that the nitrate archaea may have a significant contribution to anaerobic methane oxidation in freshwater systems. And indeed, if you look in the molecular surveys and in the databases, you can see that there are many methyl coenzyme M reductase genes of the archaea and 16S rRNA ribosomal genes of the archaea present in the database. To summarize what I've shared so far with you, it is possible to discover new anaerobes. I've hoped to have shown you that ANAMMOX bacteria make use of a hydrazine synthase to make the rocket fuel hydrazine, that we could identify the complete ammonium oxidizer ANAMMOX as Nitrospira bacteria, that Methylomirabilis oxyfera bacteria use a putative NO dismutase to make their own oxygen, and that novel nitrate-reducing AOM archaea may be responsible for a lot of methane oxidation in various environments. The final topic of today is our quest for iron-dependent anaerobic oxidation of methane. For that, we collaborate with the University of Utrecht with Matthias Egger and Caroline Slomp. They regularly go to the Baltic Sea and to the Botnian Sea to sample and measure the biogeochemical parameters in this system. So Olivia Razigraf, together with Matthias Egger, made very nice profiles of the brackish Botnian Sea sediments. What you can see is that there is a relatively low sulfate concentration in these systems, about 6 millimolars. The sulfate is very rapidly consumed in the top 10 centimeters of the sediment, so you have a very shallow, here in gray, sulfate-methane transition zone, and below the sulfate-methane transition zone, you see there is still abundant methane present and still enough reactive iron present that it could be a very good niche for iron-dependent anaerobic oxidation of methane. So Matthias and Olivia sliced the cores they obtained from the Botnian Sea into different slices, and they incubated them under anaerobic conditions with iron or manganese nanoparticles, sufficient label in methane and CO2 in case they would be autotrophs. And of course, they measured these incubations with very sensitive mass spectrometers, and they analyzed the incubations with element analyzers. Similar to the paddy soil slurry incubations, what we can see here is if you incubate the sediments as control, you see there is no liberation of 13 CO2. If you only add 13C methane to the incubations, you see there is still a small amount of endogenous electron acceptors in the systems that will yield a small amount of 13 CO2. However, when you add nanoparticles of a readily accessible iron-3 to the system, you see a relatively rapid increase in the 13 CO2 liberation. If you even then add more iron-3 to the system, you see that it continues to produce 13 CO2. We were able to wrap up this story last year and publish the results in Environmental Science and Technology. We are not the only ones hunting for iron-dependent anaerobic oxidation of methane. Maybe to tease the Americans, if it doesn't exist, you do it the American way and make it. A recent very interesting publication of So et al. Appeared, in which they engineered methanosacina into an iron-dependent AOM organism. What they did, they knocked out the endogenous methyl coenzyme M reductase gene of this methanosacina, replaced it by a copy of a MNA1 archaeon, and they were able to show that these engineered methanosacina cells were able to increase their protein content on methane and 10 millimolar iron chloride. Also a very nice publication of Sylvain Scheller in Vicky Orphan's group could show that the AOM consortia in Eel Basin, but using uncouplers and different incubations, they could also show that these AOM archaea were able to convert methane at the expense of 10 millimolars iron citrate. With that, I'm at the end of my presentation. in the last years, I have the pleasure to interact with many journalists, although Fox News might not be my favorite news station. Jenna Brenner of Fox had it completely right. ANAMMOX, COMAMMOX, oxyfera, methanoperidans, and other new anaerobic microbes could save the world. Unique bacteria hiding out in a witch's brew of anoxic water not only thrive in cold wetlands and oceans, but also chow down its ammonium and methane. And with that, I would like to thank you for your attention. Thank you very much.
Thank you very much, Mike, for an amazing seminar.
Yeah, that was outstanding.
If you want to share screen share. Okay, there we go. Okay, so if anyone in Hangout has questions? Please unmute yourself if you want to ask. Okay. We will also watch Twitter to see if any questions come in. I don't know if I've seen anything yet, but everyone loved your explosions.
Hopefully not my explosions, but
well, explosions of methane, yes. I would love to say you make anaerobic microbiology and what you've done look very easy, but we did like your, what was it, units of patience axis?
They were very high.
That was outstanding. Any questions on Twitter, send them through at Micro Seminar. Okay, so you've shown that you've got potential, you've got evidence of this iron-mediated anaerobic methane oxidation. So what's next? What's the fantasy metabolism that you're the most excited about?
I've shown you one of the slides. We still have to hunt for a iron-dependent ammonium oxidizer. Trying for 10 years already, but that's difficult not to crack.
Yeah, so I think Kerry Weber had FEMOX. They called it FEMOX, weren't they calling it that? At least they showed activity for it at some point.
There are some about 10, 12 publications about activity, but would like to know the organisms.
Sure. Okay.
What is very challenging in that type of research is that any tiny amount of oxygen will screw up the system, because a tiny amount of oxygen will have the COMAMMOX or nitrosomonas convert ammonium into nitrite, and iron with nitrite gives rise to a whole lot of abiotic reactions. So that is the challenge, to keep it as anaerobic as possible.
Yeah. So we have a question from Twitter, actually from Daniel Bond. He said that you mentioned the hydrogen synthase was about 20% of cell protein. Do you think this is slow turnover, low delta G, or detoxification?
The hydrogen synthase is a sloppy enzyme. Yes, it is very slow. The reaction is difficult. It's uphill. Hydrazine is at standard conditions minus 700 millivolts. So yes, it is a difficult reaction, a sloppy enzyme, and ANAMMOX has to make sure every molecule that is produced is taken away immediately. That also means the hydrogen dehydrogenase is a very effective enzyme. Yeah, and definitely I'm sure that Thomas in Heidelberg wants to make better hydrazine synthase. I'm sure many people in the world want to make better hydrazine syntheses. But apparently ANAMMOX had never the need for it.
Okay, we have a question from Dave Baltris. Have you been able to culture the, what was that? It went away, Dave. The one from the gill symbiont, the fish gill symbiont.
The short answer is no. So we have also, it is a recent finding, we have also not put yet very much effort in enriching the nitrosomonas or the pseudomonas from these systems. What we definitely know is that the nitrosomonas in the gills is not the same nitrosomonas as we find in the water of aquaculture. So there's quite different. There are two different types of nitrosomonas.
Okay, let's see. I've got another question here from Mingxing Jia. How do you see the fact that ANAMMOX bacteria also have the DNRA capacity?
How do I see? So if we look in the genome of ANAMMOX bacteria, we see that they, in addition to ammonium, they can also use formate, acetate, maybe also some other short-chain fatty acids. So, and especially when ammonium is low, when there is sufficient nitrate or nitrite, then I think ANAMMOX will switch over to decimatory nitrate reduction to ammonia, and in that way start to produce its own ammonia. And since there is enough nitrate, it seems to prefer the ammonium nitrite combination, but in certain conditions, under ammonium limitations, it is able to make its own ammonia. It's difficult to say how active they in the environment they are. So then we would have to find an environment with low ammonia and see how they do. We have tried it many times in reactor systems, but there is a delicate balance. If you give too much formate or too much acetate, then the denitrifiers start to grow faster. So, but yes, they definitely can do it. That we have shown with label. They are definitely capable of DNRA. And we would love to find out if in the oxygen minimum zones they may be even doing their own DNRA.
Mm-hmm.
Yep.
Mm-hmm.
Yep.
See, are there any other questions from inside the Hangout or on Twitter? Well, let's see. While we wait just one more second, what's the best way to get a hold of you? Are you willing to take questions over email or over Twitter?
Preferably not email.
Okay.
Twitter would be okay, yeah.
Okay. And he is at MSMJETTEN. I'd like to thank you very much, Mike, for taking the time to share this really, really nice talk with us. We loved all the historical tidbits and references as well, and it was a great multimedia presentation.
Thank you very much. It was a pleasure to do.
Okay. We'll look forward to talking with you more in the future.
I will be visiting ISMA in Montreal. Maybe we'll see some of you there.
Good. Yeah. All right. We'll talk to you later.
Okay. Thanks very much. Bye-bye, everybody.
Okay. Bye-bye.